Auzubillahi minash shaitanir rajim. Bismillahirrahmanirrahim. Assalamu alaikum. Good afternoon, everyone. I do hope that you all are doing well, and with pleasure, I'm uh, I'm welcoming you in the webinar series called ICCBS Talks, which we have initiated as per the advice of our very dear teacher and director ICCBS, Professor Dr. Mohammad Iqbal Chaudhry. During this series, ICCBS alumni working in the top institution of the world will deliver their lectures, share their research and academic experiences in the cutting edge fields of science and technology. Without any taking further ado, I would request my dear teacher, Professor Dr. Mohammad Iqbal Chaudhry, to introduce us with today's speaker.
Auzubillahi minash shaitanir rajim. Bismillahirrahmanirrahim. Assalamu alaikum and good afternoon, ladies and gentlemen. Very, very delighted that today is indeed a very historical day because this is a celebration of the achievements of our wonderful alumni. This institution since last 55 years have produced excellent quality graduates who are serving the top industries, top academic institutions, both home and abroad. And today's speaker, Dr. Syed Erfan Asif, is one of them. Erfan was introduced to me some 2 decades ago, and I was extremely impressed from his ingenuity and his hard work. And that has paid off because he has actually excelled in his career and has achieved lots of wonderful things, both in terms of his academic excellence as well as his contributions towards industrial development. He has been a very, very strong supporter of science in Pakistan and has been writing in support of science in Pakistan. So certainly one of the most cherished alumni of our proud institution, but at the same time, a very dear student of ours with whom we have a very special relationship. Dr. Erfan Asif has received his PhD in organic chemistry from Hussain Ibrahim Jamal Research Institute of Chemistry, which is part of the International Center for Chemical and Biological Sciences, University of Karachi. He developed his expertise in pharmaceutical chemistry and then moved on to other institutions, including Pharmaceutical Chemistry Institute in Heidelberg, Germany. He has a very vast experience of working in the quality and regulatory sectors of pharmaceutical industry, spanning 28 years in Pakistan, and then United Arab Emirates, Saudi Arabia, USA, and then in Canada. He's currently working as a senior quality consultant with Farm Inch Technology Incorporated in Toronto and overseeing various quality-related research projects at the Sanofi Pasteur vaccine manufacturing site in Toronto. Dr. Asif is author of many research publications in international chemistry journals for his research and has authored 5 professional books related to quality operation and good manufacturing practices. Dr. Erfan Asif is a regular appointee of Board of Advanced Studies and Research of University of Karachi and has been an examiner of theses as well as faculty positions. I am very, very happy that he is going to speak about a topic which is the core of pharmaceutical industry, and that is quality, because quality is all about how you perceive things and design. And he's going to talk about designing manufacturing processes which eventually lead to high quality product. With this, I would like to invite Erfan Asif for his lecture today.
Assalamu alaikum. Good afternoon, everyone in the other part of the world, and very good morning to the people who are still here in this part and they are joining this session. And thank you very much, Dr. Saab, for this very detailed introduction. I'm really honored, I'm really pleased to have been part of this, and of course the institution as well. And now, I mean, it's an honor for me to talk to everyone who belongs to this institution. Well, the topic that I decided to speak about, the main reason behind this topic was, that I know that a lot of people who are doing their research in chemistry and other part of, iccbs would probably at one point of their career prefer to go to a pharmaceutical industry. So, understanding the basics of pharmaceutical industry is very challenging when you enter into this environment, being a PhD in chemistry or pharmacology. However, many of you may even be, joining the part where you are still inventing the molecule and doing some pharmacological studies on those molecules. So basically, it's an effort or it's an attempt to establish a relationship and to enhance the confidence of the inventor of the molecule and the people who prescribe the molecule to a patient, and a pharmacist, a retail pharmacist who is actually, selling the product to the patient. So it's enhancing the confidence level of all those people that whatever you are inventing or you are doing the pharmacological study about and what you are prescribing to a patient is actually serving the purpose that it is meant for. So basically, this is the basic of today's talk. Unfortunately, we have been allocated a small time slot, around 1 hour or less than an hour. But this topic actually really needs at least 2 days that I can talk about. Anyways, I will try to encompass all the basic information for many of you who probably have an intention to join the pharmaceutical industry after their PhD. I will try to encompass all the information and all the basics in as little as 45 minutes. And we'll see if there is anything else that we can still share. So I would like to share my screen. Do you all hear me clearly? Is there any interruption?
No, it's clear.
Good, great. Okay. Okay. Do you see my screen? Quality by design in medicine manufacturing, knowledge-based talk to bridge the gap between inventors, clinicians, and bulk manufacturers. So you must have trust and confidence on the people who are at the end of the line and providing the product that one of you have discovered, or some other people who have prescribed it, and the other people who are actually selling it to the patient, that it's the right product. So how do we design the quality in the medicine? Where do we start from? Okay, so this is the basic, good manufacturing practices in pharmaceutical industry. We also call it GMP. And GMP comprises of certain factors like hygiene in production, quality management, suitable facilities and qualified personnel. Let us see what it is all about. Why is this important and critical? What is the significance of GMP, the good manufacturing practices? Well, a system for ensuring that products are consistently produced. Obviously, this is the purpose, right? That what we are producing are actually meeting the quality standards that have been set prior to the manufacturing of the product, prior to the selling of the product. It is designed to minimize the risks involved in any pharmaceutical production that cannot be eliminated through testing the final product. And I always give an example to the people to make it clearer, to make them understand what it is all about. If you are buying paracetamol, you are sure that you are buying paracetamol and the pack has paracetamol. It does not have anything that would lower down your blood pressure or it will do something else. So it is actually paracetamol. And what is GMP? GMP refers to the good manufacturing practices regulations promulgated by the US Food and Drug Administration under the authority of the Federal Food, Drug and Cosmetic Act. GMP is also sometimes referred to as current CGMP. That is what CGMP is. 5 factors of GMP. People, obviously, it starts with you. You are the one who is actually making it. So procedures, you always follow a procedure to make anything. Products that you are using and you are making. Premises, the area, the plant, the building, the room where you are actually making all the processes happening. And processes, of course, itself. The manufacturing process, the procurement process, the engineering process, the R&D, everything. Now let us describe the people first. How do you, stand here as one of the most important factor in GMP? They have to be qualified, means qualified for the right purpose, experienced. You may have some experience, you may not, and if you are not having any experience, then you are trained. If you are a fresh graduate, then of course you are not experienced in GMP or in pharmaceutical industry. So then you are provided training or a combination thereof. You could be qualified and trained, you could be experienced and trained, you could be qualified and experienced and trained all. Procedures. Let's talk about the procedures a little bit. There have to be an approved procedure. It's not that just anybody can write a procedure and then follow that. It has to be approved. And by saying approved means approved by the top-notch authority of the company, regulatory body, the R&D persons, and obviously the quality people, the most important ones. It has to be simple, precise. If you're asked to mix something for 30 minutes, you have to write that it has to be mixed with for 30 minutes at a speed of 50 RPM, not just a rough idea that, okay, it could be 20, 30 minutes or 30, 35 minutes, or maybe for half an hour. It has to be precise. Controlled. The procedures have to be controlled. Not anyone or everyone can make or can write a procedure or can make any change in the procedure. It has to be controlled. What you ask for a copy of an approved vendor list? What do we mean by that? You are going to make a product and you're going to buy some primary materials. So can you buy it from anywhere? The answer is no. You have to buy it from a qualified and reliable sources. And what do you mean by qualified? It means you have tested them, you have audited them, you have made them go through certain procedures where after which you can claim that yes, they are qualified. Their quality and efficacy, the materials, quality and efficacy is proven and tested. They have to be cost effective. There is always a business aspect in everything, in every business. Pharmaceutical is no exception. So if you are making products, besides working for the healthcare or humanity, there is still a business aspect in the whole idea that so you have to make sure that whatever you are buying and you're going to make some profit, so the material has to be cost effective. Availability. How are you going to make sure that the material that you are going to buy is always available? I mean, the availability of the product depends a lot on the availability of the primary material. So if the primary material is not available, you cannot make the product. Premises. Besides looking aesthetically beautiful, the premises needs to have certain other qualities. For example, the area where you are making the product needs to have such a highly qualified and effectively designed HVAC system that could avoid cross-contamination from one room to other room. Why? Because if you are making cephalosporins or penicillins in one of the room, you would not like the same dust particles to go into the other one where you are making brufen or where you are making some cardiovascular drug. because you know the adverse effects of penicillin and cephalosporins, and if it goes to and it goes, it can create contamination to other products, which is not meant to be an antibiotic. So it can lead to adverse effects. Environmentally controlled, obviously it has to control the temperature and the humidity where a product can be manufactured in the best possible manner. Qualified and validated manufacturing and analytical equipment. Now you would come across this word, qualification and validation, or qualified or validated, for methods, procedures, equipment, even facilities. So it is again, each and every segment has a broad meaning in pharmaceutical and needs many hours to talk about. You cannot really encompass, but to tell you in a very simple way is that when you prove a process, an equipment or material that it can produce the same results every time, there are certain procedures that you go through and then you prove it, then okay, the method, the procedure, the equipment will give you the same results every time, then you call it's qualified. But it's not just one line that I can really explain this. Okay, here we are still talking about the 5 factors: process, manufacturing process, analytical process. Here you can see that, okay, during the packaging, the product is coming in. Suddenly we see that some part of raw material is also coming from the packaging line. So we don't want to do that. So the process has to be validated. Again, the word validated. Approved and written instructions. The instructions, each and every step has to be clearly written and approved by quality, regulatory, and of course the top authority of the company. Qualified operators. You would certainly not want anyone who is well trained in operating a mixer to operate a tablet compression machine, because he does not know really. So the operator has to be, he does not need to be only an operator, he has to be a qualified operator. And obviously he needs to have the certain training, the specific education, and obviously on-job training. Validated testing methods. All the testing methods, again, I'm using the word validated. Testing methods are validated so that you are sure of producing the same results every time you test the process or the material using this particular testing method. 10 principles of GMP. Now we have gone through the basic factors, 5. Now we are going through the principles of GMP. Defined operating procedures and work instructions to establish controlled and consistent performance. The sentence itself is self-explanatory. So, and I have been talking about this when we have spoken about a few points in the previous slides, that the procedures have to be very clear and the work instructions. Availability of approved SOPs. Now, what are SOPs? SOPs are one of the very basic or fundamental, tool in pharmaceutical industry. So no matter what you do, no matter what step, how small the step is, there has to be a written procedure of that step. And we call it a standard operating procedures. Adherence to written procedures. So not only that we have the approved standard operating procedures, but we have to make sure that we adhere to that. SOPs and work instructions are available for users to reference, so making the availability of the procedures easy. Prompt and accurate documentation of work for compliance and traceability. This point will also be covered under data integrity, that whatever work you are doing, so you have to promptly and accurately document the steps that you are working on so that you can trace it and you can make sure that compliance is being made. Data entered is recorded directly in the database at the time of entry. Very important. These days, data integrity is one of the most talked about point in the pharmaceutical and regulatory and quality environment. And so a lot of work is going on to ensure that data integrity is maintained throughout the process. Proof that the systems do what they should through validation, again, the validation. Instrument calibration and sensitivity measurements and records can be managed through LIMS, Laboratory Information Management System, and are easily accessible for validation purposes. Properly defined and designed system and equipment. So equipment are an integral part of this entire procedure. Facilities must be designed to prevent contamination. We spoke about it in the previous slide. Microbial, particulate, or cross-contamination has to be stopped, has to be avoided. Next is properly maintained facility and equipment. Instruments and equipment can be flagged as unavailable if they are out of maintenance or calibration. So it is very important that if we find some equipment is out of calibration, we immediately tag it as out of maintenance, out of use, or whatever necessary way of addressing the issue is with the particular industry. They have to put that tag on the instrument or the equipment that this is our service, so that you may not accidentally get into that and you start using it and then produce a bad product, or you produce an inaccurate results. Define, develop, and prove job competency. Prevent individuals carrying out a specific task if they do not have a valid training record. If you cannot operate an HPLC, you should not be operating it, or you should not be trying to operating it unless you have the specific training. Protect products from contamination, very important. Quality and environmental monitoring functionality is specifically designed to monitor possible contamination at a specific location and sampling points on an ongoing basis. You won't believe how extensively the environmental monitoring is carried out during a process, for example, of liquid manufacturing, or especially when a trial manufacturing is going on. So how you are monitoring the microbial or the particulate matters, in the environment, and what are the limits. So it's very, very exclusive part of GMP to protect the products from contamination. So when you take a vaccine or you take an injection for any other product, for any other purpose, you make sure that the liquid is going into your blood, into your body, is not carrying any microbial contamination or any particulate matter. Build quality into products. That is the basic slogan of GMP. You don't really have to prove or test the quality of the product after they are manufactured. No, you rather build quality in the product. And how do we do that? This is how we are doing it. Perform regular audits to ensure compliance. Audit is, of course, a very important factor. So after a brief introduction to GMP, we go to GDP and GLP, good documentation practices and good laboratory practices. See, so there is nothing like that. I mean, a choice. Shall I begin in the top left corner or somewhere in the middle? Or there are so many choices. No, there are not many choices. There are just one choice, and which is written and approved, unanimously agreed upon. Good documentation practices. What is it? You see this, I'm not disorganized. I know exactly where everything is. The newer stuff is on top and the older stuff is on the bottom. So nothing like that works in pharmaceutical. To ensure the integrity and reliability of data, what do? GDP lays down the guidelines for preparation, recording, and correction of data. Maintenance of the records throughout the life cycle of a document. There is always a certain period of time when you say that a certain batch, when it was manufactured, you can always go back and check the data. You can always go back and check whatever the process was and how the process was carried out, even after 5 years. So there is a timeline that you cannot destroy the batch records. And all the years, I just thought you were very proficient. Similar to the GMP, to help the industry to establish consistent practices that will minimize the risk of misinterpretations, errors in communication, and ensuring great product quality. So everything ends up with product quality. You see, he does not know zilch, but he makes the best coffee in the business. We can't do that in the pharmaceutical industry. In good laboratory practices, it's again a system, which deals with the process and conditions that is considered to be mandatory with a view to ensuring quality and correctness in the results produced by a laboratory. Now we see that there are many elements of GLP, good laboratory practice. What are those elements that we need to? You can think about it. Okay, you say that, okay, we have made the product following very strict procedure, the process, and we were having qualified people, validated equipment, approved vendor from where we bought the product and the material. So it should produce the desired product with the perfect quality. So what do we do in laboratory practices? Why do we need to do that? But there are certain elements in laboratory as well. If you have made a product and you do not test it with a good analytical method, you cannot prove to the auditor or to any regulatory body that the product was actually 100%, or it was up to the mark, it was meeting all the specific standards. So you have to have analytical methods validation, what method you are using to test the product. It has to be validated. Equipment, well, validated equipment, calibrated. If calibration is required every day, then you are actually calibrating it every day. If the calibration frequency is once a week or once a month, you are actually following it. But this is always defined by the manufacturer of the equipment or the seller of the equipment, or on the manual of the equipment, we find out that, okay, how sensitive the measurement is, how sensitive the equipment is, and how frequent the calibration should be carried out. Reference standards. Now, in every product, you are going to test what? The active pharmaceutical ingredient. For example, if you are taking a tablet of, the painkiller, the easiest example is like paracetamol. So, you have to make sure that if one tablet is claiming to have 500 milligram of paracetamol, that it should have 500 milligram of paracetamol, at least 99.9 or 98% to 102%. There is always a window, from 98 to 102%, or in some cases even 95 to 105%. But your goal is to make sure that it is close to 100%. But how do you test it? So you need to have a certain reference standard. If you are working with the SPLC, then what are you measuring it against? So reference standard is very important. And how good your reference standard is, that is another question. Sample management. So if you are going to test a sample of Brufen, you are sure that it is Brufen, this is not paracetamol, but this is not losartan potassium or Pozar for any cardiovascular product. You know that so sample management is very important. Another big area where you have to have qualified people, trained people, approved procedures and processes. Stability analytical methods. I don't know how many of you are aware of this term, stability of the product. It simply means, and I'll explain it later, that how stable your product is. And if you're claiming it that the shelf life is 2 years, will it still remain stable at the end of 2 years? So there are methods to test it and to prove it. Microbiological methods, obviously, if this product is a sterile product, then how we are going to test this is a sterile, or if some of the antibiotics are actually tested with using microbial assay method, then you need to have the microbiological methods, approved methods, and obviously the culture media to test them against. Okay, this point number 7 is very interesting. What does that mean? If you're testing a product and it fails and you see, oh, the product does not look good. It has maybe 85% of paracetamol instead of minimum 95%. How sure you are that it's not your mistake and you tested it incorrectly? Or your equipment that you used to test the assay was calibrated or qualified and did not do any malfunctioning? Or it was a genuine failure of the product. During the production, something happened and maybe a few kilograms of paracetamol was lost during mixing, or they lost some powder and it did not get mixed up. And then of course you are getting a lower amount of paracetamol or whatever the active pharmaceutical ingredient is. So how you are going to find out? There has to be a very thorough procedure for investigation of a failure. Calibration program, another big area, usually taken care of by not only the laboratory people but also the maintenance department. That they have a schedule. They have the list of all the equipments in the laboratory or in the production area, and they know exactly when they have to go and do the calibration. Okay, how the laboratory facility is. We cannot just make a laboratory in one of our, any room. It has to have certain facilities within the laboratory where you're going to put the acids, how you're going to, there are like fuming hoods that you need to have, and you have to make sure that is where are you going to use the poisonous so HVAC system, how effectively HVAC system is working in the laboratory. Does it have the for health and safety incidents to deal with the health and safety incidents if somebody gets burned or some acid kind of like displaced, then how you're going to deal with that? So laboratory facility has to have all these arrangements in place and effectively used. Products and annual reviews. You make a certain product, maybe 10 or 15 batches in 1 year. At the end of the year, and you just look back retrospectively and see that I've made 15 batches, and all these batches made in a similar way, produce the same results. Is there any kind of like deviation or any kind of like change in the process? So you go and do a data review. We also call it product annual reviews. So for each and every product, there is an annual review program, and then you see that, okay, what was the change from January to December, and from the batch number 1 to the batch number 15 or batch number 10. So where is the curve going on? So you have to, so you will be aware of the if there is any change, slight change, and you did not notice at the time of manufacturing or the time of testing, that okay, in the beginning I was getting 99.9% or 99% and above, but at the end I see that the results are still meeting the specification. It's still within the window of 95 to 105%, but yeah, like since last 3-4 months, the batches we made was somewhere around 96%, 97%. Why? There is always a question, why? You cannot challenge that okay, this is still meeting the specification. Yes, it is, but is it going to be, if the trend continues like that, what is going to happen in the next 3-4 months? So something is wrong with the process, something is wrong with the material. I have to check with the vendor as well that did you make any change in your process, how you are producing your raw material. So it is very important for a annual review. So that will ensure that you do not suddenly end up in a big disaster. Oh, the product is failing now. So you can take the necessary steps or necessary precautions before you end up with any kind of like failure. Now we talk about quality control and quality assurance. You might have heard about this terminology so far. I'm not sure, but when we talk about, and what is the difference? and quality control. Well, in pharmaceutical industry, this is how the quality people are actually considered as. Okay, I have a very, interesting definition for quality control, and it was like in the beginning of my career, one of my quality director actually once used this term, and I really loved it. And he actually explained that quality control is basically was a reactive approach. We tested after we produced. We checked after produced and packaged. So in manufacturing, quality control is a process that ensures customers receive products free from defects and meet their needs. Okay, but what if you, at the end of the process, at the end of the manufacturing steps, and after packaging and after testing, you find that the product is actually not meeting the specified limits or the quality standards? All the batch down the drain. Quality assurance is important and critical. It all began when my call was monitored for quality assurance purposes. So this is just a joke. What is quality assurance? It is a proactive approach. So there comes the terminology quality by design. So you actually build the quality within the product. This is very fascinating terminology that is used in pharmaceutical industry, the regulatory by regulators, that how you are going to build the product in the quality rather than you testing it after it's manufactured. You have to create the atmosphere and the environment where you can get the desired product, where you can produce it. In the pharmaceutical industry, quality assurance is essential for ensuring that pharmaceutical products are manufactured to a safe and consistent standard. So this is how the terminologies were actually introduced, or they came into being. GMP or quality by design, or all those terms like validation and calibration, facility and training, experience. To actually, these are all factors that is contributing actually towards the quality assurance or quality by design, where you are going to, using all these factors, you are going to build the design, the quality by design in the product. The stability of the products and material. Also a very interesting terminology, quality after release. How can you say that once you have released the batch in the market, the product is already in the market, already being sold, and we claim that we are still controlling it. So we are controlling it, we are doing the quality even after it is released by using this stability. So the purpose of stability testing is to provide evidence on how the efficiency and integrity of a drug substance or a drug product, including its packaging, vary with time under the influence of a variety of physical, chemical, or microbiological changes, such as temperature, pH, humidity, light, and radiations. Well, in other part of the world where the temperature normally goes up to 40 degrees centigrade, you might have once forgotten the product that you bought from a pharmacy. You may have forgot it in your car, and for maybe one day, and that was like when the temperature went up to 45 degree, and you did not notice that. But that may impact the product, that may deteriorate the product a little bit, but if you continue doing that, the product may lose all its efficacy within no time, and then you say, hey, well, I think that, I was using this medicine for whatever reason, and it's not actually effective, it's not really worth helping me. So you may go back and claim to the manufacturer that your product is not meeting the specification. But then the manufacturer has his own data. He's creating the stability chamber by making sure that he continues testing the product in whatever conditions he's selling the product. If it is minus 40 degrees centigrade and I forget the product in the car outside, you know what is going to happen with the product. So stability, this is how stability helps the manufacturers to ensure that the product is still meeting the conditions, the quality in the prescribed conditions, in the conditions that it has been actually recommended to store. Okay, so the next, again, I have used maybe less than a couple of minutes to describe what stability is. They're all actually big topics, lots of things that could be talked about. Validation is just something that we can speak on about maybe for 1 or 2 days. What is validation? What type of validations are there? Process validation. What does everybody states? So process validation is defined as the collection and evaluation of data from the process design stage through commercial production, which establishes scientific evidence that a process is capable of consistently delivering quality product. Process validation involves a series of activities taking place over the life cycle of the product and process. So each and every product, when we make it for the first time, after the research and development have done their part, they have actually, established the recommended standards and specified limits and the processing steps. Then there is always a scale up. Let's go and make a commercial batch. So when you go to commercial batch for the first time, you do some extensive testing, some very stringent and very aggressive and challenging sampling and testing of the product from more than the steps that are actually recommended in the manufacturing processes or the instructions. So it's not just that, you are testing the product after it is completed or the product is manufactured or already packed. No, but at every step, we challenge the process and we create the worst case scenarios and then test it and then prove it. So we do several batches. The magic number is 3, but there are certain companies who do surveillance batches as well, as they continue for next 10 batches and do the same thing. So by creating, or collecting data and generating lots of, historical evidences and data, then you are now, confident enough that each time you use this method, you use this process, your product is going to, be the same every time. Creating an evidence trail to show that an action, process, or system leads to a consistent and reproducible result. Equipment validation. So we talked about process. Now we're talking about the equipment. The equipment I'm going to use, a tablet press or encapsulator to make the capsules, or a mixer, a big mixer, what type of mixer, each and everything has to be. First, when you install it, you qualify it. It has been installed according to the manufacturer's recommendation and the manual. And you have all the necessary requirements, the electricity or utilities and everything. So operational qualification, does it operate the way it has to be? And then the performance qualification. So whatever process I'm using it for, it is actually doing the performance the way it has been designed for. So again, there are just 3 points, but trust me, big topics and a lot of discussion needs to talk about the installation qualification, operation qualification, and performance qualification. Brief, definition of IQ verifies that the equipment has been installed in accordance with the manufacturer's recommendations in a proper manner and that all of the instruments are placed in an environment that is suitable for their intended purpose. Operation qualification, the action of demonstrating that process equipment and an ancillary system works correctly and operates consistently in accordance with established specifications. And then performance qualification. Assurance that the process continues to comply with established requirements and consistently performs as intended for the selected application. Means when I'm actually using it for the product processing, it is actually giving me the desired product. with the specified quality standards. Cleaning validation, another very critical part of validation or the manufacturing process or GMP, to ensure that a cleaning process of an equipment or facility removes chemical and microbial residues of the active, inactive, or detergent. You just made, again, maybe a cardiovascular product, and the next batch you are scheduled to produce is a painkiller. You do not want someone to use a painkiller having some traces of a cardiovascular molecule. You don't want to do that. It's not recommended for a patient. So cleaning validation has been put in place to establish the environment where you make sure that all the chemical and microbial residues of the active, inactive, or detergent has been removed. Ingredients of the product manufactured previously, and the microbial attributes. So that is what cleaning validation is all about. Well, again, we can talk about it the whole day. Criteria that the cleaning process has to meet: no more than 0.1% of the normal therapeutic dose of any product will appear in the maximum daily dose of the following product. So how do we prove it? We actually test for the previous product. In the product that we have, so we collect the rinses, we take the swabs of the equipment after we cleaned it, and then we test it. And knowing that what would be the next product. So we know exactly the next product would be Brufen or paracetamol, and the previous product was losartan potassium. So we know exactly that how, and for this, even for this purpose, that your equipment has to be, your equipment has to be so much sensitive that it can detect if there is any trace amount is found in the sample. It has to be, the detection limit would be, qualified to actually detect this much of the amount. So you see how strict it is. And no more than 10 ppm of any product will appear in another product. No quantity of residue should be visible on the equipment after cleaning procedures are performed. Other critical steps in GMP are planning API vendors approval, a huge topic. I recently published a book on this topic from CRC Press, USA. Packaging materials vendor approval, very important, and you would probably be thinking about why. So there are products which are light sensitive, so you need to have the amber colored bottles for this, or there are certain kind of like aluminum foil that has to be used when you're using the blister. Testing and approving the raw materials, stability testing. We have discussed about all these things, identification of degradates in stability indicating methods. So when we are doing stability, then we also have to have some specific methods that could analyze the degradates. After 1 year, the API in the product is being deteriorated. So what degradate it is producing? This is actually identified when the molecule is in the process of invention. One, it has been, the product, not the molecule. The product has been invented, and then of course it is being manufactured in the R&D laboratory. Then it's their part, it's their job to determine after a certain time what type of degradates are actually developing in the product, and do we have methods to find out what those molecules are, what those degradates are. So the stability indicating methods are very critical as well. Validation of analytical methods, we talked about it. Routine and periodic calibration of instruments. Preventive maintenance plan for every equipment, engineering department. Okay, so but I know what data integrity is. Do we all really know what data integrity is? I'll give you a brief description on this. Data integrity is protection of data from unauthorized and unaccountable changes. is defined as maintenance and assurance of data consistency and accuracy throughout its life cycle. I'll just go through quickly because I know we don't have much time. Many people have a lot of things to do. So data integrity is simply represented with this terminology ALCOA, ALCOA. So it has to be attributable. You know that who made this data. If Mr. ABC has done the data, has written the data, it has to be clearly identified that it was him. It should be legible. So very particular about this. We just cannot write it in our own style. There has to be very clear instruction that okay, it has to be legible, contemporaneous. Okay, I'm doing it now, but I will make the data entry maybe tomorrow or during the tea break. No, I have to do it quickly now. Original, so the data has to be original. Accurate, obviously. There is no doubt about it. Okay, if you make a change in the process, in the system, in the procedure, in the equipment, any change, and we don't really know about it, we have not done some practice about it, and we have not documented it, we have not assessed it, we will end up in a disaster like that. Can you see that? Okay, there is a small change. Red bag has the sandwiches. Green bag is your parachute. So this is a disastrous situation if we have not really taken care of the change. So change control management, what it is all about, change control. It's a monitoring system. It's a tool to review and analyze the effect of a change. If I'm going to change the turret is a part of the tablet compression machine where you have the die and punches and you actually hit the powder to compress it in a tablet, in the shape of a tablet. So if you're going to make a change in that turret, the entire machine is the same, but you're just going to change the turret because the turret is broken. You still have to go through the change control management, and you have to review and analyze what could be the effect on the product which was already validated on this machine with this particular turret. I'm going to change the turret. This is just an example. So change control is again, as I repeat, is a big topic. To determine the need for corrective action to assure that the system retains its validated status, that yes, I know that system was validated, but I'm going to make a small change in it. I'm going to change the process, not just the equipment. It could be a change in the process. I'm going to change the vendor of the raw material. It's a change. How it is going to impact the process, the product quality and its efficacy and its validated status. We have to review it. We have to thoroughly review it, and we have to determine what is the effect and how we are going to mitigate the risk. Prevents changes that could adversely affect product quality. That is what change control is. We just cannot make a change on our own will that, oh, let's do this process this way, it will save some time. Assess and monitor the impact of the change. Okay, in a nutshell, GXP, that we usually call it because it stands for GMP, GDP, and GLP, is the basis of QBD, the quality by design. If we follow the GXP, we can produce the quality within the product. It ensures quality, safety, and efficacy of the finished drug product, keeps the facility audit ready, so even if FDA can come anytime. When in USA, FDA can come anytime, but in other countries, they have to give a prior notice. But they have the authority to rate in a pharmaceutical industry if they hear about some kind of activities are going on and is not under control. So they can go anytime within the USA. But in other part of the world, if the facility is FDA approved, then they have to provide, give a prior notice, and then you can be ready of that. Well, that is the end of the talk. Actually, well, I just tried to give you people some kind of like confidence on the product that you people are, and maybe some of you will be ending up as, I mean, choosing your profession. I'll stop the sharing, and if you have any question.
Thank you, Dr. Khan. It's wonderful, very informative lecture, and very much appreciated. We do have a few questions in the chat box. If you can open and read, otherwise I can also read for you.
Yeah, sure. I can go through these questions
Yeah.
now. GMP information, very good question. If we sufficiently employ process management technologies for real-time monitoring of process parameters, which governs the critical to quality attributes of the finished product, is it allowed to avoid finished product quality control testing, at least up to some extent, test only selected parameters? Okay, yeah, that's a very good question. Lakshan Vijaya Bandra. Well, yeah, well, process analytical technology is something that was introduced around like 20 years ago, I guess. And at the time of introduction, it was being thought of. It was like considered that, yes, there should be a process that, I mean, by using it, you may stop the quality control testing. However, I think it's not all the, I heard, I mean, so maybe 15 years ago that Pfizer probably had started doing it, but it's still like a lot of budget is required to control each and every, each and every product and doing the PAT for all the products. So maybe it's just, budgeting things, or because it's like each and every equipment has to be, having that particular UV, I mean, instrument installed into it to monitor each and every tablet, each and every process. So it's just the budgeting things that is stopping. But yes, obviously, because then every batch will be a validation batch. So if the company can, afford that much amount to invest, yes, obviously, I mean, regulatory bodies will not be having any issue with that. FDA had not actually clearly, declared that, yes, we are going to stop it if you use PAT, but definitely if you use PAT, it will stop you from doing, and you can prove it to FDA or any other regulatory bodies that, okay, yes, you're using PAT, and each and every batch is actually validated batch. It's not just the 3 batches. I hope I answered your question. I'm asking this question because PAT enabled real-time batch release will have significant financial gains in a business perspective, but how far this can be scientifically allowed? Scientifically, there is no problem with that. It's just the regulatory body have to be agreed on that. I mean, based on the data that you are generating and what you are submitting to them. So it's all, I mean, entirely up to their satisfaction that how much you can satisfy their demands and their needs. Okay, my question is, what parameters are generally considered for the validation of manufacturing process and how long it takes for this? Okay, if I correctly understand, Dr. Farnaz, your question is that what parameters are generally considered. I probably have mentioned during the talk that, okay, each and every challenge, each and every process, you start from the not mixing, even dispensing, and then you go to the mixing of different ingredients or different excipients. And then finally, when you do the mixing of the active pharmaceutical ingredient, and then what you do, what type of temperature, what temperature you're using to dry it, if it is a wet massing or if it is just like different type of, mixing process are there. I'm not sure if you are aware of it, probably you are. So then you challenge each and every, but the most important and the critical process is when you complete the powder mixing and it is ready to go for compression or encapsulation. Or if you talk about the liquid manufacturing, then you are doing the multivitamin product and then you have mixed all 10 or 12 type of vitamins ready. The fat soluble are separate, the water soluble are separate, and then you're going to mix them. So it needs a lot of like things like temperature or pressure or environmental conditions. So, or if we are talking about, for example, the mixing, then it's a huge blender where you're using like 1,000 kilogram of powder, then how sure you are that in 1,000 kilogram this blender has each and every corner has the same quantity, same amount of active pharmaceutical ingredients. So we just test the powder, we take the powder, we make more stringent specification to challenge the result and the process, and then you go through the tablet compression or the encapsulation, and then you do extensive sampling with every half. For example, if the total period of compression is like one day, like 10 hours maybe, you are doing the sampling every 1 hour, every half an hour. It all depends on you, how much data you want to generate to satisfy yourself and the health regulators. So the parameters are basically each and every critical step. So first you have to find out what are my critical steps in the process. Once you find the critical steps, then you know that these are my critical parameters and this is what I'm going to challenge and I'm going to sample and test. Did I answer your question? Okay, Dr. Hidayat Hussain, how do you use the importance of the cost of drugs, lowest possible price in good manufacturing practices? I mean to keep the price of the drug low. Well, that's a business question, Dr. Hidayat. Well, by saying cost effective, obviously you have to make sure that what product you are going to, if you are going to make a product that for that, I mean, the API is available within your country's region, and you still want to buy it from some other company abroad, it will definitely cost you a lot. So you are encouraged to find a source that you can, you cannot leave the GMP behind. You still have to keep it embraced, but then you have to find that, okay, how can you make the cost effective? But if you follow the quality procedures, you are actually stopping your product from failure. You are avoiding rework, you are avoiding a reprocessing. So by doing this QBD, by following the GMP strictly, you are actually saving a lot of cost, retesting cost, reprocessing, reworking. And obviously you have to find that, okay, what would be the the price effect, I mean, cost wise, where you can find the the material cheaper than other companies, than other manufacturers. So it's a business decision, but you can definitely, I mean, make sure that yes, you can do the, I mean, by the cost, buy the material at a very effective cost.
I think these were the all questions. I'm sure there must be many with other participants, so they may either direct their questions to me, or they can directly contact you as well. Thank you so much, Dr. Erfan, for sparing your time and waking up this early for this lecture. We are very much delighted to listen your experience, and it was such a wonderful experience.
I'll be more than happy to answer all the questions if you can send it directly to me or through Dr. Hina. And I'm always available. LinkedIn is a way where I can always be reached, and I always check my messages. So if there's any other question you want to ask, feel free to contact me through Dr. Hina or directly on my LinkedIn. And thank you very much, Dr. Hina, and of course, our dear Professor Dr. Iqbal Chaudhry, to giving me this opportunity to speak to the people who belong to my alma mater. And I'm very happy. Thank you very much. I'm honored. It's very nice talking to you all. Thank you very much.
Very much appreciated. And Professor Iqbal, since he was, you can see he was on at the airport. He was
I could
he has very, he always speaks high about you. Thank you so much, everyone, for your time. And inshallah, we'll come again with another ICCPS alumni talk. Thank you, everyone. You all have a good day.